Welcome, and thank you for joining us today for the webinar on recruitment strategies for rare cancer research studies. My name is Jennifer Leidenberger, and I will be your WebEx host. I'd like to make a few comments before we get started. All lines have been muted upon entry and will remain muted for the duration of the webinar. Please submit your questions throughout the presentation in the chat panel and select Host from the drop-down. We will ask the questions on your behalf during the Q&A portion of the webinar. A moderator will facilitate those questions. If you need to view live captioning, please refer to the Media Viewer panel. Please note this webinar is being recorded. And with that, I'd like to turn it over to Dr. Lisa Galicchio to introduce our first speaker.
Thank you. The first presentation will be given by the Hairy Cell Leukemia Registry team of Michael Griever, Leslie Andrisos, and Mirella Angelina. This team has led the development of the Hairy Cell Leukemia Patient Data Registry, which they will present on today. Dr. Griever, I'll send it over to you.
Okay. Well, thank you. Thank you for the opportunity to discuss our international program in the study of hairy cell leukemia. Hairy cell leukemia is a rare form of chronic leukemia initially described in the late 1950s. Over the past 60 years, tremendous progress has been made through physician and other investigator collaboration, resulting in marked benefit for patients living with this disease. On this slide, the major features of this disease are presented. Because of extensive disease in the bone marrow and spleen, patients are subjected to the effects of bone marrow failure. Infection presents a serious challenge to the use of standard chemotherapy. The goal of therapy has been achievement of a remission while avoiding the negative impact of therapy on blood counts and the immune system. The average survival for patients with hairy cell leukemia was 4 years up until the late 1980s. Pentostatin and cladribine were developed during this time. These agents achieve a high complete response rate that is durable. In 2007, we were challenged to identify which aspects of patient management would potentially improve by working together as an international team of investigators. The Hairy Cell Leukemia Research Foundation worked with physician experts to develop concise summaries of the existing knowledge regarding this disease and to focus on plans to address unmet needs of the patient population. Approximately 26 centers of excellence were recognized to provide such expertise. And on this slide, you can see the description of what constitutes a center of excellence. It's important to note that the team of experienced individuals who have dealt with patients with this disease are engaging consultants from other disciplines that are quite pertinent to optimal management. The teams meet regularly to outline specific treatment plans, and they're available for providing consultation to other physicians who have less experience in this rare entity. An international group of disease-specific leaders worked together to construct a reliable source of information. Through the Hairy Cell Leukemia Foundation, patients could engage with experts to obtain expert opinion regarding their case. In addition, the Hairy Cell Leukemia Foundation sponsored research for junior investigators as well as for senior investigators. The research was peer-reviewed. Each year, investigators developed a scientific agenda that was designed to address specific unanswered questions in the disease. The Hairy Cell Leukemia Foundation joined forces with the SAS Foundation to sponsor these peer-reviewed research grants. They targeted questions of importance to the patients and their physicians. The Hairy Cell Leukemia Foundation has had approximately 10 international annual meetings to share progress and to interact directly with patients and their families. In addition to the annual scientific meeting where the science and the results of the research is shared, the annual patient seminar is conducted in conjunction with this meeting, and this enables patients and their families to meet the scientists and the clinicians working on their disease. Furthermore, this seminar facilitates patient-to-patient interaction, which is very important when you're dealing with a rare disease. We have identified approximately 26 centers of excellence from North America, Europe, the Middle East, and Australia. Patients can request expert opinion of direct importance to their individual case. The Hairy Cell Leukemia Foundation has sponsored many events. They've developed expert guidelines and published these for patient management. They've sponsored research and patient educational activities to improve the life of those who are impacted with this rare form of leukemia. Each year, the foundation sponsors a patient seminar where the investigators share their research progress and meet the patients and their families. Examples of this are illustrated on this slide. And then the Hairy Cell Leukemia Foundation has funded a very important patient data registry to secure clinical information on this disease. Drs. Andritsos and Angelina will expand on this international effort.
Thank you, Dr. Griever, and thank you to the organizers for the invitation to present our work. As Dr. Griever said, I am joined by Dr. Marella Angelina, who has been a key partner in this project. We would now like to describe the Hairy Cell Leukemia Patient Data Registry, including how patients are recruited and enrolled, and how we're using this to conduct research. The Patient Data Registry project actually began as a request from Hairy Cell Leukemia patients who were frustrated by the lack of information regarding hairy cell leukemia outcomes and complications. The creation of the registry required the collaboration of hairy cell leukemia patients, investigators, and biomedical informatics specialists. The overarching goal of the project is to collate searchable research-grade data. We would like to capture patients from around the globe to ensure diversity of epidemiologic patterns and also clinical practices as these vary by region. Ultimately, the long-term goal is for this to serve as a platform not only for outcomes-based research, but also for therapeutic clinical trial data collation and ultimately translational research. I would like to take a moment to describe the registry software architecture as it has evolved. Originally, this was a federated architecture with no centralized data storage. All data was stored locally at participating sites, and this ensured that each site had control over their own data. The data was not visible to investigators outside of each institution until a query was performed, at which time de-identified data was merged by the registry software and could be downloaded to an Excel file and analyzed. The major concept was that the data would be mainly extracted from the electronic health record and updated continuously, and that there would be minimization for the need of manual data entry, although there is a mechanism for this if it is needed. However, we had significant challenges with data sharing agreements, and many of the institutions would not allow the software connection, and also we became aware that many sites did not have an electronic health record. Therefore, in order for more sites to be able to participate, we are currently moving toward a centralized model. The original hairy cell leukemia patient data registry software was actually developed at Ohio State and is still here. So this has been updated and reconfigured to function as a centralized database for the registry. Unfortunately, all data will have to be entered manually so that we can avoid the contracting issues around the electronic transfer process. but this ultimately will expand the reach of the registry and allow more sites to participate. So patients are recruited to the registry via several different mechanisms. One is they can be consented at a participating site at the time of an office visit, and this is obviously at sites that have an IRB-approved protocol and have had deployment of the registry software. In addition, patients can enroll to the registry via the Hairy Cell Leukemia Foundation web portal. Dr. Griever previously showed this slide, which has the link to click to the data registry portal, and our IRB protocol includes permission to advertise and recruit patients on the Hairy Cell Leukemia Foundation website. Patients are either self-referred to the website, i.e. they find it themselves, or they can be referred by a treating hematologist to participate in the registry. We do have a flyer to advertise the registry, and our IRB protocol includes permission to advertise by way of this flyer, and this directs patients to the Hairy Cell Leukemia web portal or directly to Dr. Angelina, and these are currently being distributed by treating hematologists. This is a screenshot of the Hairy Cell Foundation data registry entry portal. Patients enter their contact information, and a member of our team contacts them to complete the informed consent and enrollment process. This is our registry flyer, which, as you can see, directs patients to both the foundation website and also directly to Dr. Angelina and our registry email address. Registry participation is fairly straightforward. Patients need to sign informed consent and sign a HIPAA consent, and also provide authorization for release of medical information. And then our team goes to the institutions and sites where care was provided and retrieves the medical information. Data is updated annually or anytime upon a major change in the patient's health status. We currently have 4 sites actively participating: The Ohio State University, Royal Marsden Hospital in the UK, University of Rochester, and MD Anderson. We have a number of sites that will be coming online, including the University of New Mexico, University of Southampton, UK, Cancer Care Manitoba, and Memorial Sloan Kettering. This slide shows our current registry participation by site. To the left is the number of patients who have been consented, and to the right is the number of medical records that have been entered into the registry. And as you see, since the registry opened, we've had a steady increase in enrollment year by year, and hopefully over time we'll soon have enough patients enrolled in the registry that our observational studies will be powered to give us significant results. The web portal has proven to be a very important mechanism for patient recruitment to the data registry. We receive inquiries from the U.S. and around the globe on a regular basis. So far, we have consented 199 patients to the registry with acquisition of 154 medical records and upload of 140 medical records. Unfortunately, we don't yet have the web portal set up to enroll patients from outside of the U.S., but we are working on this. This map shows the web portal participation by state. The green squares show the Hairy Cell Leukemia Centers of Excellence, and the red dots show patient enrollment, with larger dots representing larger numbers of patients. You can see that there are large areas of the country with no center of excellence, and so the web portal is a very important mechanism for patients to participate in the patient data registry, and it shows that patients are finding the Hairy Cell Leukemia Foundation website. This map shows the locations of our international inquiries and makes it clear that patients from around the globe are seeking information about hairy cell leukemia, and that we need to continue working to make the registry available to international patients who may not live near a center of excellence or have access to any expert opinions about their disease. The registry has enabled us to develop a number of research projects that I would like to briefly describe. These projects were prioritized because they were either requested by hairy cell leukemia patients or because they were seen as critical next steps in the advancement of the understanding of the disease. So the first study is a quality of life study in hairy cell leukemia. This was requested by hairy cell leukemia patients during one of our patient seminars, as quality of life has never been systematically studied in hairy cell leukemia, unlike other hematologic malignancies. This is a survey-based study, and the data from the surveys is linked to clinical data in the patient data registry, and we can correlate changes in quality of life with clinical outcomes such as relapse or need for treatment. The second study is an update to the risk stratification profiling in hairy cell leukemia. This has not been performed since 1982, and since this time there has been enormous changes in the diagnosis of hairy cell leukemia and molecular testing. And so we felt that it was important to update this to give patients information about their expected outcomes. The third study that we're doing is development of a novel scoring system for diagnosis of hairy cell leukemia. This project arose when people were entering data into the registry, and we realized that we needed to standardize our diagnostic criteria of hairy cell leukemia and its variants so that we would have uniform data entry. And so this project is ongoing and will be open at multiple sites. And then finally, we know that infections are the major cause of morbidity and mortality in hairy cell leukemia, and we felt that it was important to, as an early study, to look at the impact of infections at the time of diagnosis with hairy cell leukemia and how we're doing managing these patients. So from our perspective, collaboration is key when you're doing research on a rare disease. The success of our registry so far has been due to close collaborations of engaged project partners, including the Hairy Cell Leukemia Foundation and hairy cell patients, in addition to hairy cell expert hematologists and hematopathologists from around the world who have contributed to the development of the registry. We have worked closely with biomedical informatics specialists, as well as biostatisticians and epidemiologists to enable us to conduct research using our registry. And I would just like to say thank you to our team. Like I said, we collaborate with a large number of patients. It would not have been possible. And I would also like to thank the Hairy Cell Leukemia Foundation and the SAS Foundation for helping to sponsor this research. Thank you.
Thank you, Hairy Cell Leukemia team. The second presentation will be given by Dr. Corey Painter, the Associate Director of Operations and Scientific Outreach for the Broad Cancer Program at the Broad Institute of MIT and Harvard. She also serves as the Associate Director of Count Me In, which launches patient-driven research projects across multiple cancer types. Dr. Painter?
Hi, everybody. Can you hear me okay?
Yes, we can hear you.
Wonderful. First, I just want to thank the organizers for inviting me to present today, and to also thank the previous presenters for such a wonderful talk. It's incredible to see what happens when people start thinking outside the box, and it just makes me really happy to know that those efforts are ongoing. I think it dovetails very nicely with the work that I'm going to present today, which is a deep dive into patient engagement in this new era of social media and online presence of patient groups. So I'm going to talk about a project called the Angiosarcoma Project, which is part of a suite of projects that we've launched out of the Broad Institute and subsequently rolled out as an organization called Count Me In, that I'll talk about toward the end of the conversation today. But really what I want to focus on are lessons that I've learned along the way from partnering with rare cancer patients. And in order to do that, I have to take you on a bit of a circuitous route. I started off, I'm a scientist by training, but I was diagnosed with this cancer 8 years ago and ended up in an online social media support group where it was the only place that I could find resources. By interacting with other patients and figuring out how to carve a path forward both practically and clinically and emotionally, I recognized firsthand the absolute utility, need, and presence of online social groups in the space of rare cancer. I use that as a backbone and scaffold to come to the Broad Institute and help them think through and launch several patient-partnered research efforts in the space of genomics. And I'm going to start with the first one that we used as the backbone to then go back into angiosarcoma. So bear with me as we talk about metastatic breast cancer, which is not a rare cancer, but really is the scaffold for what I would like to talk about. So the Broad has been thinking outside of the box for quite some time now about partnering directly with patients for a lot of the reasons that have already been presented, but just to underscore the fact that most patient samples aren't actually included in research studies. 85% of U.S. cancer patients are treated in the community setting, and most patient samples and data have not been readily available for study, mostly because most people haven't been asked. There hasn't been a mechanism to include these people that are treated in community centers where research is not conducted. But we live in an era of technology and social media and cultural changes that now provide new opportunities to engage cancer patients and directly partner with them in our research endeavors. Our objective at the Broad Institute is to generate a publicly available database of clinical, genomic, molecular, and patient-reported data in cancer to accelerate discoveries in the development of new treatment strategies. And when I joined, I was brought on to a team that was studying metastatic breast cancer. The PI of this study is Dr. Nikhil Wagle, who's a Harvard oncologist and also a genomicist at the Broad Institute. And together with the leadership of the Broad, they wanted to see what it could be and what it could look like if you partnered with metastatic breast cancer patients in order to build and launch a project that would allow remote participation by having an online portal. So we started off, this is part of the lessons learned, we started off just by finding as many patients as possible, both in person and also on the internet, and Twitter, on Facebook, basically becoming very deferential to their experiences in ways that would enable us to be guided by them rather than us coming to them as the researchers. We were coming to them with them being the experts, helping to guide our understanding of their experiences with their disease, the nuances, the messaging, the things that resonated with them so that we could even pick a name for the project. We didn't have a name, we just had an idea. So with the partnership of patients, we came up with the name of this project, the Metastatic Breast Cancer Project. It was incredibly important to the people we talked to really underscore the word metastatic. We came up with this image. It was not this image from the start. It has iterated over time. We chose it so that it would not, necessarily paint any one type of person as being the person with metastatic breast cancer. We've had people tell us over time that it looked not as representative as they wanted, so we went through changes in an iterative process with patients to make sure that the way that we're presenting this resonated to the best of our capabilities. All the words, both here and throughout the website, but also through the consent process and in the way we message the data that we collect and receive, have been built in partnership with patients. So actually, before I go on, the project itself is a genomic study where we ask patients to provide consent to allow us to have copies of their medical records. We send them a saliva kit that is in a self-addressed prepaid box that comes back to us. In some cases, we'll send out a blood biopsy kit that we request a courtesy draw with instructions in a pre-filled out FedEx envelope that comes back to us, and permission to opt into allowing us to use archival FFPE tissues so that we can conduct whole exome sequencing of tumor normal matched pairs, and then couple that to the clinical data that we abstract from their medical record, as well as to the patient-reported data that we ask as part of the intake survey that patients fill out when they press Count Me In right here. So we launched this study in October of 2015, and to date we have had patients with metastatic breast cancer from all 50 states join. We have now over 5,000 women and men, because men get this disease too, that have signed up for the project and partnered with us. You can see that this is a distribution that matches the distribution of the population of the United States. So patients from the community, patients in Montana, in North Dakota, in Utah, from all over the country, are empowered to be able to contribute to our understanding of this disease. And it's a real testament to how motivated patients are to feel that sense of empowerment over their disease to be able to contribute in such a way. Patients, when you partner with them, they become stakeholders in the entire process. They see their words and they see themselves reflected throughout. And this is just an example, and this is an organic movement that happened when patients would receive their saliva kit. They were taking saliva selfies and posting it in social media, and they would post them with these lovely tributes, both about their motivations for joining, but also about how easy it is to join and that it was built by patients for patients. And each of these posts really would drive enrollment and drive other people, if they were curious about it but weren't sure that they wanted to be part of it, to reach out, ask us questions. And through that process, we met many more people and many more patients, and have had a lot of direct contact with people that we may not have met in person, but we know fairly well for their participation. We take the data and we try to deposit it anywhere we can while in parallel studying it ourselves. And so as of last week, we were able to deposit 237 whole exome sequence samples into SeaBio Portal from 180 patients. And we will release data in 6-month intervals while our computational biologists at the Broad Institute continue to analyze and look through that data in partnership with the researchers in Dr. Waglay's lab and myself and others who are invested in trying to make discoveries as fast as possible from these patients and their samples. So we wanted to explore other projects that would be modeled off of this. And my particular interest is in rare cancers, both because of my personal experiences as a patient, but because as a scientist, it's just unconscionable to me that we cannot study rare cancers effectively because there's just not a great mechanism that will allow for the acquisition of samples or the aggregation of patients or the funding that would be necessary. And so I look for every opportunity to push the envelope when it comes to rare cancer research. And when we had the success of the Metastatic Breast Cancer Project, the powers that be empowered us to then take on another cancer. And of course, I wanted it to be rare cancer for these reasons. In rare cancers, you often have strong online communities. Take angiosarcoma, for example. If you're looking for resources, you typically funnel down to one online social media support group in Facebook. There's just nobody talking about it anywhere else. And at some point in your frenzy for information, either you stumble in it almost like an energy funnel, or somebody that that's looking for information on behalf of you will stumble upon it, and they'll find a ton of other people that are alive with your disease, and you can just directly ask them about their experiences. And that leads to a very strong sense of community that is even stronger than what I've seen, I think, in more common cancers. There are smaller numbers of patients, so you have an opportunity, if you want to study these cohorts, to sequence the entire living cohort and make very rapid discoveries, because oftentimes they're understudied. And so it's a great way for young investigators to be guaranteed a paper, basically, because there's not much known about these rare cancers, and so they can turn around data into discoveries very impactfully and very quickly. So the findings from studying rare cancer patients could provide rationale for clinical management. Rare cancers often have no standard of care, and so by putting a minimal amount of effort into trying to understand them better, you could actually open up a treatment option for a group of people who just have nothing available to them. So, getting at the strong community, this is just a picture of people from my own support group where in angiosarcoma, there's only about 300 people per year that are diagnosed in the U.S., and we have about that many people that join our Facebook group each year. I went in there after the Metastatic Breast Cancer Project, and I put this post in, I think, March of 2016. I said, by a show of likes, can you let me know if you have angiosarcoma and you'd be interested in contributing your tumor samples to research? And in 1 hour, 90 living patients with this exceedingly rare cancer said, count me in, which was really amazing and great justification for going ahead and starting this whole process over, working with patients impacted by this disease to build the same type of online portal for angiosarcoma patients. Just to a little bit more about angiosarcoma. Angiosarcoma is a cancer that starts in the endothelial cells. It has the poorest prognosis of all soft tissue sarcomas. This is a Kaplan-Meier curve showing the survivability of this disease. Less than 20% of people diagnosed will survive. I myself am 8 years out. I used to track myself along this Kaplan-Meier curve, and I can tell you, it's awful being at the top and looking down that steep, steep curve. And now that I'm out many years, it's I can breathe a little bit of relief, but you never feel like you're out of the woods. And as a result, you always stay an active member of your community because you want to have your finger on the pulse of anything new that's going on. So with this community, we built the Angiosarcoma Project, and we launched it about 18 months ago. And we calved off another Facebook group so that we did not disturb the tone and tenor and support of the one group that we had by having it solely focusing on research. So we have a group, anybody can join it. I keep everything open. So if you want to see in real time what it looks like to work directly with patients in social media, you can go into Facebook and just type in Angiosarcoma Project Working Group. And you can join, or you can just, take a look without having to join. And you can scroll through the historical posts to see all of the ways that we work together with patients, caregivers, loved ones, to come up with this suite of messaging and images that has become the Angiosarcoma Project. So as of today, about 18 months in, we have 360 people that have enrolled in this study. This is, again, a cancer that gets about 300 people per year, and about only 20% of people survive 5 years. And so there's not that many people alive with this disease according to the back of the envelope statistics. So this is, for me, an amazing kind of tribute to what happens if you can properly tap into online communities that are already pre-existing. On the bottom, you have a timeline. We launched the project in January, on January 6th. That's when we got IRB approval. And by January 19th, we had the first 10 saliva kits out in the mail from patients that had provided consent. We started seeing those saliva selfies a few days after that. And not too far after that, we had our first blood biopsy kit back in-house. We started receiving med records within a month of launching. And then we launched publicly after this beta testing, and we had 63 patients join on day one. We had the first tissue arrive at the Broad in March, and then, we received our first blood biopsy kit in April. And as of today, we have 181 saliva kits, 230 med records from 145 patients. We have 66 blood biopsies back with ultra-low pass genomic sequencing already performed. We have 110 tissue samples from 65 different institutes, and we've already released our second batch of data onto SeaBio Portal, which is comprised of 48 samples from 36 patients, and we're continuing in both studies to push the envelope so that we can continue to deposit more samples every 6 months. This is just a look at what the SeaBio Portal looks like. You can access this from your phone or from your computer. You can go directly to our website, which is ascproject.org, and go to data release, or you could go straight to seabioportal.org and scroll down until you find angiosarcoma, and you can see that there's the CNVs and the amplifications and deletions, but there's also clinical data along with all of the genomic information that's coupled here for people's consideration. I want to point out here that one of the graphs that you can see here for tumor mutational burden, there is one bar all the way to the right that is kind of off by itself. Tumor mutation burden is sometimes looked at as a proxy for people that respond to checkpoint inhibition, which is something I think all of us would love to be able to understand better who does and does not respond to this incredible new paradigm for treating cancers. So seeing that we had a high number of tumor mutational burden samples, we traced that back to the patients and found that all of them in that bucket were patients that had angiosarcoma, which can arise anywhere in your body, having the head, neck, face, scalp area as the site that their cancer arose. So we hypothesized that there would be maybe it's in this area because it's exposed to the sun, and if we looked for the UV signature in the DNA, maybe we would find it, and sure enough, all of these cases also had a UV signature. We then went to the real-world data that is part of their medical records, and we specifically looked for patients that had head, neck, face, scalp angiosarcoma that went on to have metastatic angiosarcoma, who then maybe had compassionate care off-label treatment with checkpoint inhibitors and had an extraordinary response. We just wanted to, can we query the data in order to drill down, and are there patients that fit that? And it turns out that the answer to that is yes. We found two. Here's the two men. The first was diagnosed with angiosarcoma of the scalp 4 years ago, mets to his face, neck, and lung, received 8 different lines of chemotherapy that maybe he had variable responses to, but none were durable. And in 2016, received pembrolizumab, and within a few doses became free of disease, had an event that had made him stop taking it, and remains free of disease to this day on no therapy. We went ahead and sequenced his tumor, and sure enough, high TMB and a UV signature. In his medical record, the doctor wrote these words: after the first cycle of pembrolizumab, he had a dramatic reduction in the size and mass in his neck in the retrooral area. This is just amazing. I mean, we want to see this information that would be squirreled away in somebody's medical record, aggregated and out for everybody else to see, and couple it to all of the genomics and patient-reported data so that more people can have this in their medical record. The second patient is similar story, angiosarcoma of the face, metastatic, and again, this is supposed to be in 2016, received pembrolizumab and had an extraordinary response as well. Also developed an autoimmune kind of reaction, had to be taken off, and remained stable for many years after. So these are just kind of examples that you can couple the genomics and take a look at, you generate a hypothesis and take a look at the clinical information to see, are there, examples that could, support that idea, and then could you take those examples and then turn them out into either publications or scientific conference presentations so that the rest of the world can know in order to have an impact on the clinical care of patients. So we've since launched two other projects, one in metastatic prostate cancer and one in gastric and esophageal cancer. And all of these projects are, constantly working with patients in their own community, learning the nuances of what works and what doesn't within these different patient communities. And I think kind of one of the lessons in the take-homes is that it's really important to know your audience and to know the people that you're working with, because what works for some may not have the same message that will work for others, and people's motivations and their willingness to share and or even join the study may change, and it's incumbent upon us as in the research spheres to really drill down and figure out what that is for each different patient community. This is just a picture from 3 of our launched projects showing all of these different people that are proud to participate in research, that are, showing the rest of the world what it looks like to be somebody who has cancer, and most of the time in these pictures, terminal cancer, but smiling because they're contributing, and being empowered because they're contributing. I've got a question here. Am I supposed to take the question now, or are we holding questions?
We're going to be holding questions until the end.
Wonderful. Okay, I'm just finishing up with the last slide then. So based on these experiences, we have come together with we have formed a nonprofit called Count Me In, which you can check out at joincountmein.org. This is a initiative that's being stewarded by the Broad Institute, the Emerson Collective, the Biden Cancer Initiative, and the Dana-Farber Cancer Institute. And our goals are ambitious. We want to launch these types of projects in all major cancers in as many rare cancers as possible, if not all rare cancers, over the next several years. It takes a team. This is not, this is not inclusive of everybody, but I do want to thank all of the patient partners that have made all of this possible, and Dr. Nikhil Wagle and Eric Lander and all of the other visionaries who have helped ensure that we can do this and be well supported. It's been an amazing experience to work with so many people on this. And with that, I hand it back.
Thank you, Dr. Painter. And I'll remind everybody on the webinar that if you have questions during any time during these presentations, just to type your question into the question and answer box, and we'll ask it on behalf of you in the question and answer session. So we're going to move on to our third presentation, which will be given by Dr. Joellen Schildkraut, a professor of public health sciences and leader of the Schildkraut Program in Integrative Epidemiology at the University of Virginia. Dr. Schildkraut has been successful in utilizing cancer registries and other methods to conduct rare cancer research. I'll turn it over to Dr. Schildkraut.
Hello. Thank you so much for inviting me to speak today, and I really appreciated hearing the other speakers with different approaches than I'll be presenting, and it's all very interesting. So I will be talking today about my talk is focused on ovarian cancer, which is a rare cancer that, in my view, depicts the perfect storm of challenges in population-based research. Ovarian cancer is rare, fatal, and heterogeneous. Making this even more challenging is the ACES study that I will be describing to you today that is focused on a minority population where the diagnosis of ovarian cancer is even rarer and more fatal. By the end of this presentation, I hope to convince you that a population-based approach is a practical strategy for the study of a rare cancer. So some facts and figures about ovarian cancer. Of the 22,440 ovarian cancer cases that are diagnosed that were diagnosed in 2017, approximately 90% are epithelial cancers. So that was what where I'm focused on, epithelial ovarian cancer. They are cancers that include cancers of the ovary, fallopian tube, and peritoneum. Ovarian cancer comprises 3% of cancers diagnosed in women and 5% of all deaths. What makes ovarian cancer challenging to study is not only that it is rare and fatal, but it is also a heterogeneous disease. Therefore, enrollment of large numbers of cases is needed in conducting epidemiologic studies. There are 5 major histotypes of ovarian cancer, of epithelial ovarian cancer. It's the high-grade serous, low-grade serous, mucinous, and endometrioid and clear cell cancers. Fallopian tube cells are likely precursors of the high-grade serous cancers, where endometriosis cells are likely precursors of the clear cell and endometrioid cancers. These histotypes have been found to have a different spectrum of risk factors and molecular signatures, underscoring their etiologic heterogeneity. Additionally, 2 large screening trials have failed to show a significant benefit. Therefore, another complication is that there is no approach to early detection of ovarian cancer, and the majority of epithelial ovarian cancers are diagnosed at an advanced stage. Ovarian cancer in African American women there's a lower incidence, but an earlier age of diagnosis and poorer survival compared to white women. 1,985 African American cases and 18,250 white cases of epithelial ovarian cancer are diagnosed per year. Approximately 20% of epithelial ovarian cancer patients do not survive through the first year of diagnosis. This is somewhat higher among African Americans, where it's 30%. Although the overall incidence and mortality are lower among African Americans compared to whites, survival is worse at all stages of diagnosis. Why is survival worse? This is not well understood. It's a multifactorial process or factors that would contribute to it. Why do we observe a younger age at onset in African Americans? I hope that these next two slides would demonstrate the likely explanation. This slide shows the age distribution of African Americans and whites in the U.S. We see that the proportion of African Americans under age 50 is greater compared to whites. After age 50, there are proportionally less African Americans compared to white women due to a higher mortality rate. In this slide, we see that the incidence of ovarian cancer in African Americans compared to white women is increasingly lower with increasing age. This is largely due to the smaller number of African American women at risk in this population. So I think it's just an artifact of population distribution that we observed an earlier age at onset in African American women. So if there were a similar number at risk, the age at onset would probably be similar to whites. The next few slides depict differences in the African American population at risk for developing ovarian cancer, as well as the incidence of ovarian cancer in African Americans. These maps are useful for designing a population-based study. In this slide, the states with the highest incidence of ovarian cancer in African Americans are Georgia, Oklahoma, West Virginia, New Jersey, Washington, D.C., and Oregon. This next slide shows the proportion of African Americans residing in each state, with the highest proportion in dark blue, between 16.9% to 48.3%, seen mostly in the Southeast. In this slide, it's the number rather than the proportion of African Americans residing in each state. When planning a population-based study, this is particularly relevant because rather than incidence, the number of cases diagnosed per year is more important. This is further underscored in this next slide, demonstrating that the incidence rates can be misleading. States with the highest number of diagnoses of ovarian cancer in African American women are not the states with the highest incidence. You can see that that Oregon and Oklahoma only have about 3 and 14 diagnosed cases in African Americans per year, although the incidence in those two states was among the highest in the country. California, Georgia, Texas, and New York have much greater numbers of diagnoses and, of course, have larger populations of African Americans at risk. There are also several other factors to consider in strategic planning of a population-based study that I'll illustrate through example of my ACES study, the African American Cancer Epidemiology Study. This is the largest existing case control study of ovarian cancer in African Americans. I will review the design characteristics herein concerning ACES. And this was funded by an R01 from the NIH. ACES is a multi-center population-based case control study of African American women with invasive epithelial ovarian cancer, and we included 11 geographic regions over the course of the study, including Alabama, Georgia, Illinois, Louisiana, Michigan, New Jersey, North Carolina, Ohio, South Carolina, Tennessee, and Texas. Not only were we in a large geographic area, I just have, this would also include many diagnosing hospitals. We can only have estimated how many this would cover of at least 450 hospitals in the study regions. So the identification and eligibility of the cases included using a rapid case ascertainment system for most of the regions. I will be going into some details about this and some of the challenges we had. But eligible cases in upcoming slides, eligible cases were 20 to 79 years of age at diagnosis and, again, resided in these 11 regions. Using rapid case ascertainment, we were able to enroll cases within approximately 6 months of their diagnosis between 2010 and 2015. And one of the eligibility requirements were that they were self-reported African American. Controls were identified through random digit dialing performed by a vendor in Maine and were matched to the cases by age and geographic residence. They had to have at least one intact ovary and be African American by self-report. In this next slide, if you notice, some of the regions or states have an asterisk. These are the regions that were included in the ACEs study. 3 of the regions were SEER registries at the time of the study accrual, and that included the Detroit metropolitan area, not the whole state of Michigan, Louisiana, and New Jersey. 5 regions, Alabama, Louisiana, Georgia, South Carolina, and North Carolina, are among those with the highest proportion of African Americans. This guided our choices of the study sites at the time, and we also wanted to have a northern and southern representation in the study to achieve a distribution and representation. 2 regions, Texas and Georgia, are among those with the highest number of diagnoses. So here's some idea of what we did in terms of the procedures to enroll women into the ACEs study. First, we had to receive physician approval to contact the patient. Once we ascertained their contact information. This was mostly passive. We allowed for 3 weeks to pass. If we did not hear from the physician, we went ahead and sent a letter and a brochure to the eligible subjects. And we followed that up with a telephone call to schedule a baseline survey. We had trained telephone interviewers who performed the baseline survey and follow-up surveys, except for Michigan residents. Approval from study cases was obtained from the coordinating centers that could receive patient information in New Jersey and Texas. So this was a second layer in those 2 regions that some of the other regions didn't require before we could contact. Michigan actually hired their own staff and conducted the telephone interviews and obtained biospecimens. We also provided a website and toll-free number for subjects to address questions and concerns. Before we might have had a chance to even speak to the patients ourselves, they were able to call our office or get onto our website. The coordinating center was at Duke University, where I was at the time. We also provided subjects incentives up to $100 per subject, $50 upon completion of the baseline telephone interview, and $50 upon receipt of the DNA sample, which was obtained either. Our first choice was blood, and second choice saliva. We also contracted with a vendor called EMSI, who sent phlebotomists for a home visit to draw the blood, or at times, if requested, a saliva sample. Some of the in this next slide, I've described some of the data elements in our baseline questionnaire. This would be typical of many epidemiologic studies, including demographics, medical and reproductive history, family history of cancer, exogenous hormone use, lifestyle behaviors such as smoking, drinking, and physical activity, analgesic use, body powder exposure, perceived discrimination, social support, trust in physicians, and height and weight. We offered a short questionnaire option to convert would-be decliners to responders, and this comprised about 8.7% of our cases and 2.5% of our controls that might have not had participated otherwise. Other information that was collected from our cases and controls included a food frequency questionnaire at baseline. We also conducted yearly follow-up surveys that addressed quality of life, physical activity, medication use, social support, stress, and anxiety, amongst some other things. As I mentioned, we were collecting DNA samples from either blood or saliva, but we also obtained FFP tissue on the primary ovarian cancer diagnosis. For one thing, we had centralized path review to confirm the diagnosis, and then used additional, we are using the tumor tissue in other studies of looking at molecular signatures, for example, of ovarian cancer in African Americans, where very few of those exist, and other studies as well. We've participated in GWAS studies additionally, and we are also using these specimens in a whole exome sequencing project. Clinical data from the medical record abstraction was also obtained, including chemotherapy, surgery, residual disease, a very important prognostic factor, CA-125 levels, and complete blood cell count. Not all the data are complete. It's a very, as I mentioned, how many hospitals potentially are involved in these requests, but we've done reasonably well given that this is a population-based study in such a wide geographic region. I'll be providing some statistics on this a little later. We also update vital status from using cancer registries, and to date, we've published 22 manuscripts on this study. We've involved many of our co-investigators, have gotten a chance to get a publication, so it's been very productive. In these, the next slides, in preparing for this presentation, I took a look back at the information that of the different sites that we had, and in preparing and planning this study. So I'm just going to try to give you some idea of what we went through and some take-home messages in planning such a study. In this table, I've described the characteristics of the cancer registries that can impact our accrual. These are the sites that were included in ACEs, and we did run into some problems. For example, the Illinois registry lost funding and could no longer perform rapid case ascertainment and was replaced by Texas. We lost a northern region that was replaced by a southwestern region. It could not be helped, but again, we ran into trouble with and lost the rapid case ascertainment in Texas. So there are a lot of things we could not control. In fact, although not anticipated, about half the sites did not have rapid case ascertainment that was fully functional. And when you're thinking about a fatal disease such as ovarian cancer, this is very critical for representation on a population-based study. But some of the sites were able to compensate at some expense by going to major hospitals in the area, for example. So we had our struggles. Michigan, as I might have mentioned, would not send Duke, which was the coordinating center, the patient identifiers at all, and therefore they conducted their own interviews and obtained a blood sample using their own personnel. I think for the most part that worked out okay, but it gave me and our central group, our center, less control. From a quality standpoint, I'm not sure that we're having a problem or not, but it seemed like my interviewers were working harder and faster. So I would have preferred to have had more control over that as well. Other barriers included that there was a required pre-consent, and I think I did allude to this earlier, in New Jersey and Texas, before the contact information could be sent to Duke, where we had set up a CADI system to conduct a centralized telephone interview. In this next slide, I present the response rates by study site. The highest were in South Carolina, North Carolina, and Ohio. The lower rates are in red, and these included the 3 SEER registries, all of which had functional rapid case ascertainment. In preparing this presentation again, I've looked back at some of the information we were using to plan the study and maybe some take-home messages from that. Here I show the number of projected cases from each of the sites that we obtained in the first column from the registries themselves, and then besides those are the number of cases we actually did receive. For the most part, there was a reasonable correspondence between the projected and number of cases and the number we received from the registries. The exception was with Georgia, perhaps because they did not record the race, and this was not communicated to us at the time. So I think the Georgian numbers were probably all races combined, and that's why we see that they had projected 90 when we actually received 47. However, just out of curiosity, I went back to the NACER website, where the registries post the number of cases they accrue per year. And across the board, all of these numbers that the registries report were much greater than those that we ever saw in our study in terms of accrual. So I would just say that I would advise people to communicate, in planning the study, to communicate directly with the cancer registries to obtain more realistic data rather than rely on information on their website. This slide shows the median days from diagnosis to ascertainment and the median days from diagnosis to interview by site. Two stand out as very long, Texas and New Jersey. Both these were the two sites that required pre-consent before personal identifiers could be sent to the coordinating center. On the positive side, overall, we had approximately 4 months from the time of diagnosis to ascertainment and 6 months from the time of diagnosis to interview, which is very reasonable in a population-based study such as this, even though there was variation within these different registries. I thought this was kind of an interesting slide that we prepared, which shows that, this next slide, which shows the actual, what it's done is it's mapped out the residential locations of our cases and controls, showing that there's very good geographic coverage in many of our states except for Ohio and Texas. Since Texas had lost rapid case ascertainment, they worked more directly with large hospitals. And I think you could see how those clusters are reflecting some of those large hospitals in Texas. And this is similar in Ohio, as opposed to other statewide registries with better coverage. The next few slides show the characteristics of the ACES sites, as well as other locations that might be considered based on the number of cases, number of African American residents, and incidence rates. The green on the far left column depicts the ACES sites in this slide. It is clear that not all the desired characteristics are aligned, whether it be the frequency of cases or the proportion of African Americans or the incidence or the number of cases per year. This next slide, I did restrict it to just the ACES site so that you could see that, again, not all those desirable characteristics are completely coincidental with the sites we chose, which also says that you can consider several of these characteristics and probably conduct a successful study. The other thing that I wanted to add that I'm probably not giving as much credit as I should at the end of this presentation is the expertise and experience of the investigators at these sites that contributed to the success of the study, and I can't and also in the productivity of the study in terms of publication. So I want to. At each of these sites, there were PhD epidemiologists who had experience with enrolling cases, or, whether or not it was ovarian cancer or not, and some of them have particular expertise in the epidemiology of ovarian cancer. So in this next slide, I have a take-home message that compares the ACES study to an earlier study that I did in North Carolina called the North Carolina Ovarian Cancer Study, and another study in Los Angeles, the Los Angeles County Ovarian Cancer Study. ACES was the shortest, had the shortest enrollment period of the other compared to the other two, but enrolled 116 cases per year compared to 13 per year in the North Carolina study, which was just a 63-county study, and 7 per year in the Los Angeles study. So I would say that ACES is a winning strategy for a rare cancer. Just to give you a little bit of an idea of the other types, the success we had in obtaining other types of information or in the study, we were able to get a DNA sample on 75% of our subjects, which did require a separate signed consent that had to be mailed into the study center. Our cooperation rate among controls was 68%. We were able to obtain pathologic slides and H&E, and we sustained our own H&E on 80% of the cases. We received a food frequency questionnaire in 83% of our study subjects. This was self-administered, although when requested, we provided assistance. We were able to obtain medical records on 84% of cases, although it included some missing data. This also required signed release forms to be mailed back to the study center. And the follow-up survey after the first year of diagnosis, we were able to interview 72% of the eligible cases. In summary, our challenges were low response rates, the large geographic region that was required in multiple states, and the variation in cancer registry capabilities. 15% of our ACEs subjects were deceased at the time of ascertainment, underscoring the high fatality rate. This compares to 4% among white women in an older North Carolina study. So these are the kinds of challenges we were up against when I mentioned in the beginning of my talk the perfect storm. Controls, surprisingly to me at least, had a higher response rate than our cases, although both cases of control had similar cooperation rates. There is a lack of trust of the medical community among African Americans, which does contribute to the response rates. Again, I've mentioned that we've had to interact with many hospitals to obtain tissue and medical records. We had a long startup period to get everybody on board, another challenge. And the loss of rapid case ascertainment capabilities and the lag in case ascertainment was another challenge. And I have to say that when you have a functioning rapid case ascertainment and no pre-consent requirement, it works better. So I have to thank everybody, and that's the end of my talk.
Thank you, Dr. Schildkraut, and thank all of our presenters today for great presentations. We're going to move into the question and answer period. So if you have a question, please enter it into the Q&A box, and I will be delivering those questions to our presenters. Before we move into the question and answer session, I apologize. I was excited about moving on to our presentations, and I didn't give the full introduction to the hairy cell leukemia team. So just so everybody knows who they are, Dr. Griever is a hematologist and faculty emeritus at Ohio State. Dr. Andritos is a hematology specialist and professor at the University of New Mexico. And Dr. Angelina is a physician and outcomes researcher at The Ohio State University. So I apologize for not giving you that information before their presentations. So one of our first questions, I think, is a good question for Dr. Painter. Has your project looked at how to effectively communicate information back to patients, and looking at both the type of information and the format of that information?
Thank you so much for that question. It's an excellent question. This is something that is always a work in progress. We have and continue to look at effectively communicating back to patients what our aggregate findings are. And one of the things, we take for granted things that we think would be interesting for patients versus what they find interesting. And I'm going to draw a specific example that I came across from actually the metastatic breast cancer patients, but I think it's applicable to all patients, just in terms of not taking for granted what we as researchers think is interesting versus what patients might find interesting. When we released our first batch of data to SeaBio Portal, it's a wonderful platform, freely available through Memorial Sloan Kettering. They house anybody's data for free. It's a remarkable resource, and it's really meant for scientists. So it's not necessarily something that patients could just intuitively look at and understand. And so one of the things we wanted to do was, at a minimum, go through and put descriptors over every single data element there. And so we enlisted the help of about 40 patients to help us go do walkthroughs of the data, look through the definitions, put lay-friendly terms there, really give good descriptors around what it meant. And then we basically walked them through the data, and we were thinking that here's a visual representation of all of the different genes in the patient-reported data and the clinical data, and that patients would really love it, right? That they would want to just dive in and click through everything and try to understand, the underpinnings of the disease. And when we showed it to 40 different patients, they had responses like this. One of them cried and said, "Oh my God, I'm not alone." One of them said, "I don't necessarily need to know the specific genes. I want to see who is 40 years old and HER2 positive, and I want to see what therapies they were on." They were really looking for clinical insights. they had a whole different perspective through which they came to the data with that is completely different than our intentions for housing the data in the first place. And it was only until we started talking with them that we really understood and recognized that. And so now what we do is, we're building a new app that we can hopefully have types of data that will be high impact for patients to be able to easily click and learn about what it is we're learning, but in ways that they're interested in. And we'll build that with them. When we get to that point, we'll ask them exactly what they want to see, how they want to see it, how they want it represented. And in the meanwhile, we put out aggregate data in every social media channel, in digests that we send by email, on our websites and blogs. And then we do, we give a lot of scientific presentations at conferences. I'm at a conference right now, for example. And what we'll do is we'll do a lay-friendly Facebook Live event where whoever the presenter is will do a lay interpretation of the project and the science learned. And then that becomes a recording for the patients to be able to look at, so they can have the lay interpretation overlaid on top of the scientific learnings, and in real time ask questions. And if they don't see it in real time, then they can email us or leave comments that we can then come back and address. So I hope that gets at this question.
Yeah, thank you. For the hairy cell leukemia patient registry team, what kind of data are being collected and entered into the registry? Are there epidemiologic and health habit data being collected, or is it just clinical data?
So we are actually entering as comprehensive of a data set as we can. So we do collect clinical and health data, but we are looking at occupational and exposure histories, and very minute details of the patient's diagnosis and treatment history. So we're trying to basically put in there a complete medical record, which is why it has been so time-consuming via the manual data entry portal.
So related to that, is it difficult to harmonize data across sites for the hairy cell leukemia patient registry, or are they entered in the same format?
They're entered in the same format. There's one manual data entry portal software architecture. So everything goes in a uniform method. When we actually started the patient data registry, we met at one of our annual meetings and basically came together as a team of hairy cell leukemia hematologists and hematopathologists, and agreed upon what were the important data elements to include in our registry. And this was something that actually took a long time to get through and multiple email iterations, but eventually we did come to a conclusion about what was important to capture so that we can go back and look, and with a major focus on epidemiology, because that's been extremely difficult to conduct in hairy cell leukemia, and most of the studies are relatively small.
Thanks. Dr. Schildkraut, you talked a lot about rapid case ascertainment. And can you explain a little bit why it's important to have rapid case ascertainment when recruiting through registries?
Sure, that's a great question. The registries in many states by law are required to report cancer, but not on it may be on an annual basis, it may be on a quarterly basis, or every 6 months. Just for the registry to surveil what the incidence is, and to provide some statistics for the state or geographic region. However, if that schedule for a highly fatal disease, as one example, that where within 10 months, 30% of African Americans, for example, or 20% of the general ovarian cancer population may not survive past 10 months to a year, we would be missing those cases without rapid case ascertainment. We do miss some of them, but because of rapid case ascertainment, we probably are more inclusive of the spectrum of the disease, of this aggressive disease, and because we were able to include them more quickly. The other issue is, as the disease progresses, habits change. People change some of their eating habits and physical activity. Various things can change due to the disease and the diagnosis. And so in an epidemiologic study where you're trying to ascertain risk factors and determine risk factors, you want to be able to say, have some confidence that the risk factors or the reports in the surveys about risk factors really did occur prior to the diagnosis and didn't represent a change in habits. So it's tricky in a case control study, which is retrospective in a sense. So the most ideal case control study, you'd want to get them even, as they're being diagnosed, to prevent a lot of time lapse between the diagnosis and the time that you're in contact with the patient. So for several reasons, in terms of avoiding reporting bias and misclassification of the data you're trying to collect, as well as being representative of the spectrum of the disease, knowing about the patients earlier than would have been the normal reporting process is important, and that is through rapid case ascertainment, which has a cost, per patient cost typically in each of these registries. I can tell you, most of my experience had been with the North Carolina registry, which as time went on, they improved, and actually we would know about a diagnosis within a couple of weeks of the diagnosis where we felt it was a little too soon to contact the patient, so we'd wait a little while. But when it works well, it's more inclusive of the disease.
Thank you. So there are a number of questions across all the presentations about data sharing and access to the data in the resources that have been generated. So perhaps we could start with the hairy cell leukemia team and talk about how can outside investigators gain access to your registry, or can they at all, and what would be the procedures if they can?
So actually, our patient data registry is pretty open to investigators. So essentially, anyone can query our data. They have to submit a proposal to the Hairy Cell Leukemia Patient Data Registry Steering Committee, and the steering committee exists so that sites can actually opt out of studies if they're already doing something similar, and to make sure that there isn't overlap between studies. But the inquiring individual would need, an IRB approved protocol to conduct the research. But our intention is for the data to be available to anybody who wants to do hairy cell leukemia research. And so the steering committee is essentially one member from each participating site, as well as a hairy cell leukemia patient representative, a member of the Hairy Cell Leukemia Foundation, and a biostatistician to assess projects for feasibility. And in this way, we hope to make, the registry available to anybody who's interested to do hairy cell leukemia research.
Great. Dr. Painter, I think you touched on this a little bit during your presentation, but can you talk about data sharing for the projects that you're involved in?
I'm happy to. I love the question. Was it challenging, as though maybe hinting that other people have had some challenges getting their genomic data housed in public domains? It is challenging. So we have it currently, somatic calls in SeaBio Portal, with the process kicked off many months ago to start housing in the Genomic Data Commons. But to get to the Genomic Data Commons, we have to go through dbGaP. And to get into dbGaP, it's quite a long process that includes lots and lots of forms and back and forths, and quite a circuitous route. We are helped tremendously by the crew that is shepherding that process, and we're very grateful to them, but we're not quite there. So we have the hopes that in January of 2019, data from both the NBC and the Angio project will be housed in the Genomic Data Commons, that we'll be able to have raw genome, germline and somatic data there, likely have the clinical data still living in SeaBio Portal, but have a hashtag that will be able to be linked between the two.
Great. And Dr. Schildkraut, can you talk a little bit about data sharing in the context of your study?
Yeah, that's a little bit different because we don't have a public access per se. But ACES is part of two consortia. One is the Ovarian Cancer Association consortia, where we participated in GWAS studies where that data would be submitted to DBGAP as well through the consortia. And it was part of some funding from NCI as well, called the Game On Network, where I think we went through CIDR and I believe everything's been uploaded someplace through that network. Also, there's some minimal data set of ACEs has been contributed to OCAC, as well as the North Carolina Ovarian Cancer Study, which was a prior study. We've contributed tons of data to OCAC, where members of OCAC have access, but not the general public. Then there's another consortia that I oversee called AQUA, which is very focused on African Americans. And in that consortium, we are it's early on, but we are in the process of harmonizing data across a number of epidemiologic studies, and over time that would become more available, I think, through other data sharing agreements we have with NIH. But that would be all epidemiologic data and not genomic data at this point.
Right, right. So we have a question about consent, and Dr. Schildkraut, the pre-consent is usually controlled by the IRB. How do you get around that requirement? So there were certain sites, I think, that you said didn't require pre-consent.
Yeah, I think it was the registry IRBs, not necessarily the institutional IRB.
Okay, so the registries
seem to dictate that.
It.
Yeah, most of the registries do not. They may have their own processes. You do go through registry IRBs, though. All the registries typically have an IRB that we went through. So the North Carolina, South Carolina registry and others did not have a pre-consent requirement. In North Carolina, every cancer patient is given a flyer of some. I think it's mailed to their house, and they can opt out if they don't want to be part of a future research project. But most people don't opt out, and so if they don't opt out, then the registry permits us to obtain HIPAA, information for determining eligibility for the study, and then we get physician approval and begin the process of contacting patients. And that is typical for a lot of the registries, and the exceptions were those two, where they required first the representative from New Jersey that was on our study, the co-investigator, she had her team contact the patients first to permit that their contact information and personal identifiers could be passed on to the coordinating center. Similarly, and with Texas, and what that just does, there are just layers of delays in that. Even if the patient does permit their information to be passed on, it just means that another contact has to be made, and all these are independent factors that can interfere with actually finding the patient and response rates. Whatever additional layers you add in, additional phone calls and contact that has to be made and delays, it just contributes to, I think it has a detrimental effect in the overall response rates.
Thank you. It does, yeah, thank you. I think that answers the question. Mm-hmm. So the final question I think I'll ask of everybody, and it has to do with social media. So we'll start with the hairy cell leukemia patient registry team. Are you using social media to sort of reach out to patients for the registry? And do you think that's a practical approach to do so?
So we have been really behind on using social media. Really, our only online presence is with the foundation website. And so we know that there are Facebook groups out there, and we've discussed internally reaching out to them, but we have not yet done so. So I'm really interested to hear this discussion on this webinar around use of social media and identifying, potential participants in some of these studies, because I think that's something that we really need to do.
So Dr. Painter, do you think that social media has been underutilized in cancer research? Because it seems like you've had a lot of success.
Yeah, totally. I think I'll say a couple guiding principles that we use. In some instances, different types of social media works better than others, and especially in terms of patient privacy. That should be the number one overarching concern for anybody that wants to use social media for research purposes. I could go on for hours, and I know we don't have that much time, but if anybody wants to connect with me offline, please feel free to contact me. I'll say a couple things. One, the one guiding principle is if we wanted to, for example, reach patients that were in a closed Facebook group, we will not join that Facebook group. None of us ever want to come across as interlopers and or inappropriately spying on patients that are there for reason. It's closed for a reason. And, you know, if you're not directly impacted by that disease, you likely should not try to get in there unless specifically invited by the admin and making your presence there known and that everybody is comfortable with you in there. And so just as like a guiding principle in terms of reaching out to patients that you don't have, you know, a direct relationship with. I had a situation with the Metastatic Breast Cancer Project where I started an open group. I don't like to have closed groups for the stuff that we do because I think, you know, you run into issues of, you know, false sense of privacy. And so I like to have open groups where everybody knows it's open and they don't have to join. And I think that's key is you can still look at the stuff and you can still participate by anonymously messaging and or sending emails to us and having us post anonymously for you and therefore not feel like you're left out. But, you know, a lesson that I learned was that I didn't make it clear enough when I had the first open group for working with patients with metastatic breast cancer, even though, you know, I put it in the description. Not everybody reads the description. And so then I went ahead and you can ask questions for people to join your group. And one of the questions, like the first one is, are you aware that this is an open group and anybody in your Facebook feed can see that you're part of the group and also that, you know, and just like basically you have to check this and say yes to be part of it. But, and even though we took those measures, the tone and tenor in that group did not feel right to me. So I closed it and then archived it. And instead we went and opted for a Facebook page where it's less kind of like a back and forth dialogue with patients, but at the same time, we have opened up different, you know, avenues to communicate with those patients. So it is a little bit tenuous trying to, you know, tread that line. Whereas in angiosarcoma, that patient population is very open. They know that it's an open group. They have to answer the questions to get in, and it's not been, you know, it's not been an issue. Not everybody that's in these groups are patients. You know, it's open to the whole community, so you can have doctors and caregivers and loved ones. So just by virtue of being a member of that working group does not identify you as a patient. But at the same time, we heavily message the importance of understanding that there is no privacy if you are going to be participating in a social media chat about your disease that we can protect, you know, and so obviously we protect everybody's data once it comes in through the study, but that's a whole different thing than people being open about their disease in the space of social media. And so those are kind of just lessons learned. Twitter is an open platform completely, and so, you know, that's just, I think, incumbent on the user to know what their protections are, whereas Facebook is a little bit of you know, you can get yourself in some tricky waters, and my suggestion is to err on the side of being extraordinarily transparent, even more so than you think you may need to, so that everybody is aware of the risks.
Right. Well, thank you. Thanks for that guidance.
Sure.
There's one comment from the Hairy Cell group I just wanted to make. When we have these annual meetings and involve the patients, we've had very good attendance so far. What we've tried to do is do them in different cities, and we alternate between North America and Europe. This last one was in New York City, and they're very well attended by the patients with some family members. But the next one's going to be in Rome, and when I was talking to some of the Italian investigators who work with us, they were saying that some of their patients culturally may not be as open to coming to these patient seminars. Now, I don't know. We'll see how this goes. But in Germany, when we had the meeting about 2 years ago in Heidelberg. The German hairy cell leukemia group is extremely well organized. They communicate a lot. They have, they even have like a magazine that goes around, or a bulletin. And in the United States, there's a lot of interaction. So I think it would be interesting to see, number one, what are there cultural barriers, to talking about a rare disease, depending on where your patient population is. And the second challenge will be in some of these circumstances. When we went to Germany, for example, while the scientific meeting was conducted in English, we may have had some of the patients who had difficulty, and so if you have an international organization trying to address you can't just assume that all patients are fluent in English.
Right.
I'd like to echo some of that more in the scenario of the ACEs study. And, when you were focused on minority and rural populations, what I'm also finding out more recently, having moved to Virginia, not everybody has a cell phone. Not everybody has connection to the Internet. So if you would be possibly systematically excluding certain people if it was dependent on some of the social media and Internet connections.
That's very true. Well, we're past our time. I want to thank everybody on the webinar for their attention and active participation. And I'd really like to thank the three presenters or the three presentation teams for their really excellent presentations and insight. For everybody, we are planning several additional webinars in this rare cancer webinar series in 2019, and we will be posting information about these webinars when the details are finalized. So thank you again for your participation.
Thank you all. Bye.
Hi, thank you.
Thank you.
Bye.
Thank you.